Hello, I'm Faiza Saeed. Today we will talk about a company, Novoenzyme, in the field of biotechnology and analyze the problem they are trying to solve and also the solution they have come up with. And also being a student in the same field, biotechnology, how I will tackle the problem in a different way. Novoenzyme address major industrial challenges like energy consumption, chemical usage, and environmental pollution. Because many industries use harsh chemicals, excess water, and also high temperature for the production of materials. So, Novoenzyme has provided a sustainable way by using enzyme, microorganisms, and biosolutions, which perform the same function but in a faster, clearer, and a sustainable way. Let's talk about the Novoenzyme approach and its enzymes production workflow. First, they identify the screen. First, they screen the microorganism and identify the right enzyme which can be used in industries, and then they genetically engineer it and optimize the enzymes for the industrial production. And after that, they move to the industrial scale for the fermentation in large bioreactors, and they proceed to the purification of the enzymes and concentration of the enzyme. Then they formulate the enzyme for a specific industrial usage. Novoenzymes approach have pros as well as cons. The pros are they are innovative, it is efficient, and it is a sustainable uh, approach. Let's talk about the cons of the Novoenzyme approach, and that is high cost. Screening and engineering the enzymes, it is usually expensive. Enzymes are sensitive, enzymes are heat sensitive, pH sensitive, and they can also be sensitive to a very slight change in their environment. These enzymes are dependent on the fermentation and may have a regulatory barrier. Like different countries have their different regulatory systems. Adoption challenges may occur. Industries may be resistant to change their old traditional chemical approach. These were the limitations of a Novoenzyme approach, and as a biotechnology student, I have came up with my solutions which can tackle the same problem but with a different approach. So my solutions may be engineering microbial strains which remain alive and have this perform same type of reaction inside the process itself. So these biocatalyst, it is easily scalable, and it may have low production cost. And instead of using purified enzymes, we can use this approach. And another approach can be creating microbial consortia which perform the specific function and similar to its ecosystem. Like they will be able to directly convert the cellulose into biofuel, directly convert the cellulose in sugars, and also it will increase the amount of yield we get, and it will improve the biofuel yield by removing the inhibitors. And my another approach can be developing biosensors inside the microbial cell itself, so they can sense the changes in the industrial conditions and automatically adjust themselves for the enzyme productions. We can also immobilize the microbes on the beds so that industries can use them repeatedly for a longer period of time instead of extracting the enzyme from them. Talking about the challenges of my solution, it may have lower specificity because whole cells produce many unwanted enzymes and metabolites that are harder to control. Which reaction may occur because of which metabolite, and also it will have a contamination risk because living cells can get contaminated easily by the microbes, and it will require strict aseptic conditions. And the conditions are more complex because cells require aeration, temperature, and other controls, which usually purified enzyme do not need. And talking about the challenges we may face by using synthetic microbial consortia, are maintaining population is difficult because one specific microbial population may overgrow other population and suppress the others, and because of the production of inhibitors, other microbes may die. And it also requires high monitoring needs because monitoring pH, temperature. Talking about the challenges of small bioreactors and AI controlled enzyme expressions, it is an expensive setup, and small industries may not take this approach. And also the sensor limitations may occur because small error in a biosensors may disrupt the whole process. These biosensors may not work under harsh conditions like harsh extreme pH conditions and extreme temperature conditions, and also specialized staff is needed. Thank you.